This is Biology for Grade 9, produced by Tigray Education Bureau, transmitted by Dimtsi Weyane Tigray National and FM radio satellites and social media, and also FM Maqala 104.4.
Hello students, this is Biology for Grade 9, presented by Tsaggai Gebremetin. Our today's topic is microorganisms. By the end of this lesson, you will be able to 1. Define microorganisms and list the main types. 2. Distinguish between useful and harmful microorganisms. 3. Explain how vaccines are made and work. 4. Identify disinfectants, antiseptics, and antibiotics. 5. Describe the methods used to control microorganisms. Students, let us define microorganisms. Micro means small or tiny. Organisms means living things. So, microorganisms are tiny living things that are too small to be seen with the naked eye. To clearly see these organisms, we need to use magnifying devices such as microscopes. They are mostly unicellular and microscopic. These include all bacteria, some fungi, all viruses, some algae, and all protozoa. Students, let's discuss something on bacteria. Bacteria. All bacteria are unicellular organisms found in the kingdom Monera. They are much smaller than the smallest animal and plant cells. The common structures found in all bacteria are cell wall, made up of peptidoglycan, which is different from cellulose found in plant cells, cell membrane, cytoplasm, non-membrane bound ribosome, and DNA, deoxyribonucleic acid. Some other structures found in few bacteria are flagella, used for movement, capsule, used for protection, and pili, used for reproduction. They lack nucleus. Instead, they have nucleoid without nuclear membrane. In general, they are devoid of membrane-bound organelles like mitochondrion, chloroplast, lysosomes, etc. Bacteria are found in different shapes and sizes. Some are rod-shaped, called bacilli, singular bacillus. Some are round, called cocci, singular coccus. Some are comma-shaped, and some are spiral-shaped, called spirochetes. Students, let me ask you a few choice questions. 1. Which of the following cellular organelles is not found in bacterial cells? A. Ribosome, B. Cell membrane, C. Cytoplasm, D. Mitochondrion. The correct answer is D. Mitochondrion. 2. Rod-shaped bacteria are referred to as A. Bacilli, B. Cocci, C. Spirochetes, D. A and C. The correct answer is A. Bacilli. 3. Which of the following structures is common to bacterium, animal, and plant cells? A. Nucleus, B. Chloroplast, C. Ribosome, D. Nucleoid. The correct answer is C. Ribosome. 4. The hair-like structures in some bacteria used for reproduction are A. Capsule, B. Pili, C. Flagella, D. Cilia. The correct answer is B. Pili. Students, do you think that all bacteria are harmful to us? If your answer is no, you are correct. While some bacteria are useful to us, there are some other groups that cause many diseases. The role of useful bacteria are the following. Many bacteria are used in the making of bread, alcohol, yogurt, cheese, and vinegar. Some are used in sewage treatment and laser processing. Some are useful in the genetic engineering technology to produce insulin, hormones, enzymes, vaccines, etc. And some others play a greater role in decomposition of dead organic matters and recycling of nutrients such as carbon, nitrogen, sulfur, phosphorus, oxygen, and hydrogen, which are the main components of life. On the other hand, some bacteria can cause many diseases such as tuberculosis, cholera, pneumonia, and many other sexual diseases. Students, let me take you to a very interesting subtopic: identifying bacteria using simple staining. Students, we have already said that bacteria are too small to see their structures. However, scientists now are able to identify bacteria by using simple stain method. Stain is a dye used to color cells and tissues for microscopical examination. Different types of bacteria have different cell walls that can react with the different dyes. So, the basic principle of stain technique, the simple stain technique, had been developed by Hans Christian Gram for the first time and hence named Gram stain in his honor. Gram stain helps to see the structures of bacteria clearly and to group them into gram-positive and gram-negative. Students, let us see the steps of gram stain method. 1. You stain all the bacteria on the slide with the primary stain crystal violet, which will stain all the bacteria purple. 2. Add grams iodine as a mordant and leave it for 1 minute to form a violet iodine complex. 3. Add 95 ethanol, which acts as a decolorizer to the violet iodine complex. This washes crystal violet out of some types of bacteria, but not out of others. 4. Then rinse the slide in water until no color is left. 5. Add safranin dye as a counter stain. Safranin is a basic dye that stains the decolorized bacteria red. 6. Leave this for 1 minute and then wash off with distilled water. 7. Blot the slide gently dry and observe it under a microscope. The bacteria that are easily decolorized and so stain red by safranin are named as gram-negative bacteria. And the bacteria that maintain the crystal violet and so stain purple are named gram-positive bacteria. This method is very useful for doctors to identify whether the bacteria is gram-negative or gram-positive and decide the prescription of proper antibiotics to a patient. Students, let us have a look on the next group of microorganisms. Fungi. Fungi are groups of organisms which obtain food from dead or living organisms. Some of the microorganisms of this group are parasites. Others, such as molds and yeasts, are most useful to us. Both molds and yeasts are very important decomposers. They break down animal and plant materials and return nutrients to the environment. Yeasts are single-celled microorganisms. Each cell has a nucleus, cytoplasm, plasma membrane, and a cell wall. They reproduce by asexual budding, which means splitting to form a new yeast cells. Students, please refer to figure 4.3 on page 125 in your textbook. Molds are made up of minute thread-like structures called hyphae. Hyphae are tubes consisting of a cell wall and cytoplasm containing many nuclei. Molds reproduce asexually by producing fruiting bodies containing spores. Students, please refer to figure 4.4 on page 125 in your textbook. The third groups are viruses, which are even smaller than bacteria. They have a genetic material with few genes surrounded by a protein coat. They cannot carry out any of the life processes of organisms independently. They can reproduce only when they get a living cell. Hence, all viruses are taken as parasites. Even though we study them along with the microorganisms, nevertheless, viruses have not reached the level of organisms. Examples: rabbit virus, human immunodeficiency virus, HIV, tobacco mosaic virus, coronavirus, etc. Let me now ask you a few choice questions. 1. Hyphae are extensive thread-like structures of A. molds, B. yeasts, C. bacteria, D. viruses. The correct answer is A. molds. 2. Which of the following groups of viruses is taken as a catastrophic disease that kills people at the moment? A. HIV, B. tobacco mosaic virus, C. coronavirus, D. rabbit virus. The correct answer is C. coronavirus. 3. Which of the following groups of microorganisms reproduce by budding? A. bacteria, B. yeasts, C. viruses, D. molds. The correct answer is B. yeasts. Students, have you heard about germ theory of diseases? If you don't, we are going to deal with it now. The germ theory of disease states that any disease is caused by microorganisms or germs. Time in the past, many people followed spontaneous generation theory, which claimed that living things could arise from non-living things. But in the 17th century, a Dutchman, Anton van Leeuwenhoek, discovered a microscope with the help of which he could see bacteria for the first time. Robert Koch improved the microscope, which helped him much to see many tiny organisms clearly. But it was after the work of the Frenchman Louis Pasteur in the late 19th century that the germ theory of disease could get acceptance by people. Pasteur proposed that fermentation reactions used to make beer and wine are the results of the actions of microorganisms on glucose. Pasteur conducted a series of experiments to disprove spontaneous generation theory. He boiled brose and sealed the container tightly to protect the air from mixing with the brose. The brose remained clear. When he introduced air with the microorganisms to the boiled brose, the brose became cloudy, indicating that microorganisms are grown in it. From that time onward, spontaneous generation theory was rejected once and for all. Pasteur went on identifying the microorganisms that caused diseases in people and animals such as anthrax, rabies, and diphtheria. He found methods of weakening or killing the microbes and prepared vaccines that promote body defense against diseases. Pasteur's work convinced everyone that infectious diseases are caused by microorganisms. Students, do you support spontaneous generation theory or germ theory? If you support the latter, you are on the track of science. Students, please read the immune system, which we are going to deal with next week. Till then,